All right. So we are going to talk now about nucleotide metabolism. So this is our last biomolecule, big biomolecule. All right. So I always I really like your book starts these chapters off talking about practical applications, right? So if you think about something that affects a huge number of our population, that would be cancer, right? And so if you look at this picture here, these are two cancer cells that are like here, these are supposed to be like epithelial cells. And the epithelial cells have undergone mutations. And so now they're cancer cells. So if you think about kind of the hallmarks of cancer cells have insanely high rates of cell division. They consume a lot of nutrients and they just divide and divide and divide and divide. And they don't care about anything else. And so we can actually use that to our advantage. They are going to replicate more quickly than most of the cells in our body. And so if you have a cell that's replicating over and over and over and over again, what do they need to make lots of? DNA. When you think about replication, what are you spending a lot of energy making? That's DNA. So what do you need to make DNA? Well, you need lots of nucleotides. Right? That makes sense. So one enzyme that is essential for the production of um, are different nucleotides, particularly thymine, which is a nucleotide base in DNA, is dihydrofolate reductase. And so, if we can use our knowledge of how this enzyme is essential for the biosynthesis of our nucleotide, then, then we can capitalize on that. So, if we inhibit the thymine synthesis, well, we're going to block DNA synthesis, we're going to decrease rates of cell division, and so it'll work. So, this drug, methotrexate, why can't I write? It's a picture, didn't want to let me do normal stuff. Okay. Anyway, will it not me now? Okay. So methotrexate is the drug, and this is the drug that targets our thymidylate synthase reaction. So it's an enzyme that's required to make a coenzyme. Like we'll talk about the pathway later on. But if you inhibit this enzyme, then cells can't divide because they can't synthesize DNA quickly and they like run out of thymine. And it's really pretty cool. So you take this drug and the drug binds inside of the active site and you inhibit your cancer most of the time. But well, I mean, any cell whose only goal is to survive, whether it's bacterial or human, whatever it is, they often develop mutations. And those mutations can actually prevent the binding of the drug. And so this is an example of that, where you have something like this L22R mutation in the active site that prevents your drug from doing its job. And so the cancer cells become resistant, and it can even get really bad. We'll talk about this kind of later in the chapter that you can become resistant to even multiple drugs. And so that's a really big reason why cancer is so dangerous, and 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 why we really need to move into cancer treatment being specific to the patient and not super general like kind of we've been doing so far. So we're really going to talk about um, chemical structure of our nucleotides. We're going to talk about biosynthesis. We're going to talk about degradation because nucleotides are so fundamental in the cell. They participate in most reaction sequences inside the cell. I mean, they're just essential. And so I want to kind of remind you guys about nomenclature, right? So if we call something a nucleoside, we're talking about just the base and the sugar. If you call something a nucleotide, that's the nucleoside, which is the base and the sugar. And then we're going to add to that 1 to 3 phosphate groups. So if I have ATP, I would call ATP, y'all know these, y'all know this, adenosine 5 triphosphate. But sometimes what you're going to hear is that it's called adenylate. Adenylate is kind of like the generic name instead of being super precise with it. So just be aware that you're going to hear kind of both of them. All right, so we'll talk kind of chemical structure first and functions and that kind of stuff. So I kind of like for y'all to know sort of where we're going, big picture, before we get into too many details. And so when we talk about nucleotide metabolism, we're trying to build really big things with nucleotides, which we call the nucleic acids, mainly DNA and RNA. And so what do we use to build DNA and RNA? Those are the nucleotides, which are the sugar base and the phosphate. Where does the sugar come from? The sugar comes from ribose. Our nitrogenous bases can come from a bunch of different places. It can come from proteins, can come from amino acids. And then you also have to think about how are we going to break down these nitrogenous bases, because the nitrogenous bases can't be stored, right? They got nitrogen in them, can't do it. So if it's a pyrimidine, it kind of gets broken down into your ammonia. If it's a purine, it turns into uric acid. And we're going to talk about all this in the future. Okay, so nucleotides are really used in these 4 big, really important biochemical processes, right? And so energy conversion is like the first one that people think about outside of storing genetic information, right? Genetic information, that's our last one down here, DNA, RNA. But energy conversion, people think, okay, ATP, yeah, that's energy conversion. But we've talked about how like the citrate cycle makes GTP, right? And all kinds of different things. So it's not the only one. ATP is not the only one. It's kind of just the one most people think about. Signal transduction pathways. This is where you're starting with a cyclic nucleotide, like cyclic GMP, right? Or cyclic AMP, those kinds of things. Then you have coenzyme dependent reactions. So what are the coenzymes? We've talked about this, like nicotinamide adenine dinucleotide. We just been calling it NAD plus, but they actually have names, right? FAD, flavin adenine dinucleotide. So it's still the adenine and it's a dinucleotide. Coenzyme A is another example, and we'll talk about them pretty extensively. Okay, so let's go look at structure first. So what does our nucleotide have? Well, we have a base, and that base is either a purine or a pyrimidine. We have a ribose sugar, and we have a phosphoryl group. So if we look just at the base, this is what's going to tell us if we have a purine or a pyrimidine. And I want you to notice when we look at the sugar that's attached to the base, you see all of these have primes. So don't forget from Biochem 1 that we name them primes because they're the carbon from the ribose, not from the base. So the carbons from the sugar have the primes, the carbon from the base have no primes. Okay, and then you have some number of phosphate groups or phosphoryl groups that are attached to the sugar. So when we talk about these different nucleotides, they're different if we have RNA versus DNA, right? On C2, if we have a hydroxyl, it's RNA. If we're on C2 and we have just a hydrogen, it's a deoxyribonucleotide. And so when we talk about our different nucleotides, right, purines and pyrimidines, you should know your purines and your pyrimidines. We have 5 here. We have our 5 main ones. There are some others. We'll talk about them. But here are our 5 main ones. So why in the world do we have T in DNA and U in RNA? This would be a bonus question or something. You should know this from Biochem 1. And it's not about stability. What induces that difference in stability? That's the hydroxyl. This causes the instability in RNA. And we're going to talk about that extensively in this chapter. So it's the hydroxyl that tells me if my ribonucleotide, when I incorporate it in, the RNA will be unstable. And then if it's just the H, it'll be DNA and it'll be very stable. Okay, so stability has nothing to do with T and U. So I have T in DNA, U in RNA. So it really, the reason that we have those different bases is that it protects the cell against DNA mutations. So we know that cytosine can spontaneously deaminate to uracil. Spontaneous, happens without any input. So if we have a C that gets mutated to a U, and I also have like a normal, I'll call it a normal U or a cytosine U, how's the cell going to tell the difference between the two? How's the cell going to know, okay, this one should be there and this one shouldn't? It can't tell the difference. So if I start out with something that's a GC pair, right? My C is going to get mutated to a U, right? My G is going to be okay, and I'll go and make a normal copy of that one. But if I come over here and I take that U, I split this double-stranded apart, and I have a U, well, what's going to pair with U? A is going to pair with U. So then the next time I replicate this DNA again, I'm going to have an AT pair. I started at this location with a GC, and now I have an AT. And this will happen a lot. And so you end up accumulating a lot of mutations, and that would be really bad. So the cell has enzymes that recognize any U in DNA is just cut out and removed and replaced with a C, because the only reason U would be there is if C were deaminated. Okay, so you should know your purines and your pyrimidines. So I thought I put them on here, but I have like a little way to remember your purines and your pyrimidines. So if you look at purines have 2 rings. So it's 9 atoms and 2 heterocyclic rings, meaning you have carbons and hydrogens. And so the shorter the name, the more rings. The longer the name, the fewer the rings. So that's how I remember purines and pyrimidines, generally their structure. And then if I look and I'm like, gosh, which ones, A's, C's, T's, G's, U's, which ones are purines? The purines are pure as gold. Pure as gold. So that's how I remember purines. And then pyrimidines. So what do you do when you get a pie? You cut a pie, but really it's pyrimidine, right? P-I-R-I-N, pyrimidines. So you cut your pie. So C-U-N-T. So that's how you kind of remember it. But what I want you to kind of add to your notes is that these 5 bases come from precursor molecules. So the precursor for purines is hypoxanthine, and then the precursor for pyrimidines is orotate. And we're going to look at the structure and we're going to look at all that. But I want you to know that there aren't just these 5 bases. There are other bases, and even in addition to the hypoxanthine and the orotate, but they're just not common, and so we don't talk about them very much. Okay, let's see. And then you do need to study the structure. They're very, very similar. So if you look at adenine compared to guanine. There are very few differences. So in adenine, right, we have a nitrogen base, where in guanine, it's a carbonyl. And then you also have a nitrogen group that comes off right here. But otherwise, it's pretty much the same, right? You lose that double bond there, but pretty much pretty similar. So then if we look at C, I want you to look at C to U. What's the difference between C and U? Well, C, we said deaminates to U. So if I look at that place where it gets deaminated, it gets replaced by a carbonyl. The amine group gets replaced by a carbonyl, which then means you lose the double bond and then you pick up an H, right? Okay, so those two are pretty similar. And then if you look at T and U are extremely identical. The only thing that's different is that T has a methyl group right there. So you should definitely know your structures of your bases. Okay, so everybody, when you think nucleotides, most people think, oh, ATP. So we're going to talk about the role of just one nucleotide in a bunch of different things, right? So ATP makes it really easy to do that. Why did I underline that one? Okay, so energy conversion is your first like go-to when you think about it, right? So if you had to give an example and say, okay, what are the 4 major biochemical processes that use nucleotides? Well, you'd say energy conversion, signal transduction, coenzyme dependent reactions, and genetic information. That's what we got from the very beginning of this this chapter. But you have to be able to explain that, right? So if you look at what they're showing you for energy conversion, well, what's happening in this energy conversion? What enzyme is that? That's glucokinase. Glucokinase. So what does glucokinase do? It uses ATP, right, in order to phosphorylate, to add that phosphate group to glucose, right, to trap it in the cell. So now we have glucose 6 phosphate and it's trapped, right? So we can use the energy in ATP to do things in the cell, to convert different molecules and store it and all that kind of stuff. Okay, signal transduction. Oh, here, this one we talked about. Cyclic AMP, right? So ATP can be converted into cyclic AMP. And this is an example that comes from a G protein coupled receptor. We see that right there. And that G protein coupled receptor uses cyclic AMP as its secondary molecule, which activates pKa, which does whatever downstream effect, right? So you're explaining how it's important. Let's see, what's the next one? Coenzyme dependent function, right? So this is NAD, nicotinamide adenine dinucleotide. Why do we need this? Pick an example. It doesn't matter which one you pick. Mine one that I'm going to pick because I love our lab is lactate dehydrogenase. Lactate dehydrogenase reaction requires NAD as a cofactor inside of it. So you can pick any reaction you want to kind of describe it. And then genetic information, right? A's and T's, base pair in DNA to store our genetic information, right? Okay, so now we get to the good stuff, right? So when you think about making these nucleotides, they're pretty big. And the bigger something is, the more costly it is for the cell. So if you talk about making it from scratch, we call that de novo synthesis. So de novo synthesis is complex, requires lots of energy. It takes 9 high energy phosphate bond equivalents, right, in order to make ATP. That's a lot, right? So we would like to really avoid that energy expense. And the cell is fantastic because it uses a salvage pathway, right? We say the body is such a great recycling system. So when you think about where you're getting nucleotides, Yes, you're going to eat stuff that has nucleic acids. So you're going to get some in your diet, but you're really not going to use them as a source of nucleotide bases. You're really not from your diet. So most of the nucleic acid that we have to get rid of and would use in a salvage pathway are going to come from degradation of nucleic acids that are inside the cell. And so if you think about, gosh, what kind of nucleotide containing nucleic acid is going to have to be broken down often? That is RNA, right? RNA has a very, very high rate of turnover. Once you make DNA, it kind of stays. So you don't really want to mess with it, right? And so when we look at this pathway, we're going to start kind of at the top. And we're going to start with an RNA molecule. In the RNA molecule, we have these little squiggle lines to indicate that it goes longer on either side. Okay, so if we want to take this RNA and we want to break it down, what kind of enzymes break down nucleic acids? They are endonucleases. Endo meaning you have this long chain and you got to kind of cut it up from the inside. You're not going to cut from the outside of the strand. You're going to cut from the inside. So that's an endonuclease. And what are you going to get out of that endonuclease? You're going to get these little short couple of nucleotides long, and we call those oligonucleotides. Now, if I want to break up those oligonucleotides, I'm going to use a phosphodiesterase. What does a phosphodiesterase mean? It is going to break phosphodiester bonds using water. So if I do that, I'm going to result in nucleotides. Nucleotides having the base, the sugar, the phosphate. And in this case, we're saying it only has one phosphate group because it comes from an RNA molecule. And we know an RNA molecule has that sugar, phosphate, sugar, phosphate, sugar, phosphate. And then the bases go point in the middle. So you're only going to have one phosphate. But if I want to recycle this nucleotide, I can't immediately incorporate it in. It's too low of energy. So what I have to do is I have to go through kinases and I have to go through 2 rounds. And depending on which nucleotide you're making, you're going to use a different nucleotide as an energy source. And so we just call it NTP. So this is a nucleotide triphosphate. So A's, C's, T's, G's. And so once you phosphorylate it, you are going to make, and your book has this error, so I'm pointing it out, you're going to make a nucleotide triphosphate. So be careful with that. So now I have NTPs. I started out with NMPs. Now I have NTPs. So now if I want to incorporate this into a growing strand of RNA, I'm going to use a polymerase. I'm going to make a polymer. And so that's how we make RNA. It's very, very similar to make DNA, but it's just not used very much. You use this salvage pathway a lot for nucleotides. And so this is assuming I don't want to change anything about the nucleotide. I'm happy with which one it is. That's the one I need. So it's all good, right? So you don't have as much of an energy investment. You are investing some energy. You are, but not too much. It's not bad. Okay, so now what happens if I have my nucleotide, right? This is my NMP, and it's got one phosphate group and my sugar and my base. And I don't want that particular nucleotide. I have too many CTPs and I need Gs, whatever it is. You go through a similar process, but in this case, we have to break the base off of the sugar. So the first step is to go through a nucleotidase. The nucleotidase puts removes the phosphate group from the sugar. So in place of where you had a phosphate group, you now just have a hydroxyl. And so once we have that structure, which we now call a nucleoside, because it's just the sugar and the base, now we go through phosphorylases where the base is replaced by a phosphate group. So now we have a phosphate group coming off of this is carbon number 1. Because if you notice up here, this is 1, 2, 3, 4, 5. You start out with a phosphate group on carbon number 5. Carbon number 5 loses the phosphate group. Then carbon number 1 loses the base, but then gains a phosphate group. So the base gets released. So now I've separated them and I have a sugar and I have a base. So let's track this base and see what happens. Well, now maybe I have kind of this pool of different bases. So I take the one that I want and I have to add it to, right, a sugar that's primed. So we call a primed sugar phosphoribosylpyrophosphate. Do you have to know this whole name? Yes, you do. This is an extremely important molecule. So it's phospho for this phospho, then a ribose group, and then pyrophosphate. So it's phosphoribosyl pyrophosphate. We call it for short PRPP, but you do need to know the whole name. So because this has a pyrophosphate attached to C1, this site on the PRPP is very high energy. And so what I can do is displace the pyrophosphate with a base and make a nucleotide monophosphate. And then my pyrophosphate, right, gets broken down into 2-pi, which is a negative delta G, which drives the reaction. So that's great. Okay, so what makes this sugar that has a pyrophosphate on carbon number 1, and where does that come from? So let's take a step back. If we had our nucleoside and we split off our base from our sugar, I have a phosphate group on ribose. So where can I get ribose if I don't have this, right? You got to kind of think about what happens in the cell if my nucleotides are low. Like where do I get a sugar from? Well, we know the pentose phosphate pathway can supply us with a sugar. In plants, it primarily comes from the Calvin cycle, but we don't really care about plants, so I don't care that you know that so much. But for us, we get it from the pentose phosphate pathway. So we start out with the sugar that is phosphorylated on carbon 1, and we go through a mutase reaction, and that mutase reaction moves the phosphate from position 1 onto position number 5. 1, 2, 3, 4, and 5. Okay, so then you go through this key enzyme, this really, really, really important enzyme that I've highlighted and starred, and it's called PRPP synthetase. This is highly, highly regulated. And you do take one ATP molecule and you are going to pyrophosphorylate carbon number 1 with 2 phosphate groups. And so this requires the equivalent of 2 ATPs worth of energy, because in order to regenerate this, you have to go from AMP, which is 1 phosphate, to ATP, which is 3. So it's really an equivalent of 2 ATP. So you have to prime that sugar to accept the pyrophosphate so that then the base can be attached where this pyrophosphate now is. So PRPP is important in the salvage pathway, and it's really also important for de novo nucleotide synthesis. So just realize that we're going to be talking about PRPP again. All right. Okay, so my advice to you is to try to draw these 2 salvage pathways. One is for the nucleotide salvage and one is the base salvage pathway together, because they happen in the cell together. And so just they're the two pathways on this slide and this slide. And when you're trying to do this, try not to look at your book as much as possible or your notes. And I want you to kind of think about what's the common metabolite, and that should help to lead you in the right direction. So pause it and try. If you need a little help, the common metabolite are nucleotide monophosphates. So if you didn't get it yet, that might give you a good starting point. That's kind of like your middle molecule. So go ahead and pause it and try it if you haven't done so already. So here's my drawing, right, of putting these two pathways together, where my key molecule here is my nucleotide monophosphate, right? That's the base, the sugar, and one phosphate group. And then I drew both of my pathways, right? And I included my generation of my PRPP and things like that, right? So just take that and try to put it all together. All right, so now we're going to talk about purine metabolism. So before we talk about purine metabolism, I kind of hate that your book jumps straight into it. So what I am going to do is I'm going to talk to you about both purine and pyrimidines, because they have really essential differences. And if you kind of have a heads up of what's going to happen, I think it makes a little bit more sense. So remember your purines and your pyrimidines. Purines are pure as gold, and for pyrimidines, you cut a pie. So let's talk about this. Your book gives you these two like kind of simplified pathways. So what I want to do is I want to kind of focus just on the purines to start with. So our purines, things like, let's see if I can get my laser pointer back, AMP, GMP, right? So what we do with this is we create a common base. And the common base right here is hypoxanthine. And this base can be converted into the base for GMP or the base for AMP. So hypoxanthine is the common purine base. And it's built in quite a few reactions that we will talk about. But basically, you start with the sugar and you build the base on the ribose. So the ribose is like the scaffold, and then we start to build onto that sugar. And so the first nucleotide that we make is not AMP or GMP, it's inosine. So inosine has a hypoxanthine base attached to a sugar with one phosphate. So that would be inosine. The phosphate's on carbon number 5. So it's inosine 5' monophosphate. So you do have to learn those. You do have to learn those. And then once we have the IMP can be converted to AMP, or IMP can be converted to GMP. Okay, so now let's kind of switch gears and talk about pyrimidines. And we know our pyrimidines, right, are the CUT. I guess I put them in the wrong order, but that's okay. All right, so what we do is here, we're not starting with the sugar, right? We're going to combine carbamyl phosphate and aspartate, and we're going to build the common base. So that common base that we are building is called orotate. So orotate is built first. It's a 6-membered ring. And then we attach this built orotate to our PRPP molecule. And once we attach the orotate base, what we're going to make is oridine 5 monophosphate. I ran out of room. Sorry. So then our OMP gets converted first to UMP. Then we can take that UMP, convert it to CTP and to TMP. And it takes a bunch of reactions, and we'll kind of talk about that. But I just want you to know where we're going with it, right? Okay, so let's come back up to this real quick. So we have a common intermediate. We talked about that. Our common intermediate is going to be our inosine 5 monophosphate, and that's what we're going to make. An inosine monophosphate contains the hypoxanthine. Hypoxanthine, we said this is the base, right? This is our, I went too far. This is our nucleotide. So just kind of keep those things straight in your brain. And it occurs, the synthesis of this nucleotide occurs in 2 stages, because remember our purine is 2 rings. So if we need to make 2 rings, we're going to have 2 stages, right? And so just remember that this hypoxanthine base that has 2 rings is going to be built directly on the PRPP scaffold. I think that's how to spell scaffold. Anyway. Okay, so we talked about that. So let's talk about our stage 1 of purine biosynthesis. And we're going to add to that in just a minute. Okay, so let's do this first. This is our goal. Our goal is to make inosine monophosphate. So the question is, how different is inosine monophosphate to the products that we want to make? Like, once we get this pathway done, how much more work are we going to have to do? And so if you look at the structure, so all I'm going to do is copy this. Copy. And you should be able to know your structures. So let's kind of compare, right? So this is what we start with. We're saying that each of these sugars, these nucleotides, GMP and AMP, come from IMP. So if we start with it, what modifications do we have to make in order to get to the bases that we want? And so if you look at GMP, if we look at the structure of GMP, how different is it from what we have? Well, not too different, right? What do we have to do? Well, we're going to have to add a nitrogen containing group here, right? And what else do we got to do? I think that's about it, right? Not too bad. GMP is pretty similar. How about AMP? What are we going to do here? Well, in this one, we're going to replace this carbonyl group, right? We're going to get rid of that carbonyl group. So let's kind of get rid of it. And what we're going to do is we're going to add here another amine. So we have an NH2. We're going to lose this hydrogen. Did I have a... Yep, we're going to lose that hydrogen. And then we're going to gain a double bond. Ooh, it's too short. Okay. We're going to gain a double bond there. And what else do we have to change? That's about it, right? So do we have a whole lot of differences between our IMP, GMP, AMP? Not too much, right? Not too much at all. Okay, so let's look at kind of how this whole process works. So you're going to start with a sugar, right? Because we said, remember, when we're building this, that we're going to start with our scaffold, that's our sugar, and we're going to build our rings on top of it. All right, and we got to build 2 rings. So this is just stage 1. We're starting with just stage 1. So we're going to start with a ribose 5-phosphate, and we already talked about this step, right? We said this is a very, very important enzyme. This enzyme is PRPP synthetase. So it takes our ribose 5-phosphate, and it's going to take us some ATP because we're doing a pyrophosphate. So this is going to actually take us 2 ATP molecules to actually do. Okay, so we now have PRPP. So our PRPP gets converted into what's called 5-phosphoribosylamine. And I don't care that you know the names of the rest of the intermediates, right? I kind of just want you to know where the different pieces come from. And kind of a little bit of regulation. So let me talk to you a little bit about regulation. These 2 enzymes, PRPP synthetase and maybe that was too dark, but our glutamine PRPP aminotransferase, this is really important because we're going to be replacing our pyrophosphate with something. Well, what are we replacing it with? We're replacing it with an amine group that comes from glutamine. Because remember, glutamine has our 2 nitrogen containing groups, we cut one off, we get glutamate. So these two are our kind of really, really highly regulated steps. And it's mostly feedback inhibition. That makes that work. So you do kind of have to know where in the pathway we're regulated. And you think about this, when do I want to be regulating this pathway? I want to be regulating it early before I invest too much into it. So our first nitrogen, you have to know where it comes from. Our first nitrogen comes from glutamine, right? And that amine replaces our phosphate, our pyrophosphate that's on carbon 1. Okay, now we go through our next reaction. Our next reaction, we need to add that nitrogen is kind of like the beginning of our ring. And so we're going to add an amine group and a carbonyl from glycine. So our second amine group comes from glycine. Then we're going to go through another reaction, and the informal THF, that's donating a formal group, and that's why we have our another carbonyl that's attached to the nitrogen, and that's where that kind of comes from. Okay, so then we're going to add another nitrogen containing group. So I need something else that has nitrogen. So our third nitrogen that we're adding comes again from glutamine. So at least you have a few duplications, right? So we're adding our amine right here. And then we're going to go ahead and close the ring. And to close the ring, that's going to require some ATP. Anytime you have to cut off a nitrogen containing group, you need to use an ATP. So gosh, we're using a lot of ATP. Let's kind of backtrack. So we need 2 here. We're going to need our ATP here. We didn't have to use it here because we had PRPP. And so that pyrophosphate kind of like ran the reaction. Okay, and then we come down and we have another ATP that we're going to have to use up here, and another ATP. Oh my gosh, this is getting expensive, right? And so by the time we get to the bottom, we're not even done. This is just one ring. This is one ring, and we need 2. So far, how much ATP have we spent? We spent 1, 2, 3, 4, 5 ATP. Whew, getting pricey, getting pricey. Okay, so now we got to go on to stage 2. So in stage 2, we're going to take a carboxy group from bicarbonate, right? So not only do we need nitrogens, but we also need carbon backbones. So bicarb, but again, this is going to require an investment of ATP. Then we're going to take a nitrogen containing group and a carbon skeleton from aspartate, and that is also going to take, guess what, ATP. Oh my goodness, we're getting expensive, right? All right, and then we're going to go through and we're going to start replacing over here our carboxylic acid, our carboxylate groups with amine groups, right? Then we're going to keep going and we're going to donate another formal group. When we donate another formal group, if my pen would work. Why is my pen not working? Uh-oh. See if it's not paired. Oh, I got energy. Okay. So here's our formal group. Come on. All right, I'm gonna have to pause the video. All right, we're back in business. So from the formal group, we're going to get another carbon and an oxygen. And then we're going to go through one more step, IMP synthase. And when we go through IMP synthase, we're going to finish closing up that ring and getting our 2-ring system going. So the kind of question is, well, how much ATP have we used here? Well, I know in stage 1, I used 5, right? And then I've got 2 more ATP. So that gives me a total of 7 ATP that I had to expend. This is why salvage of nucleotides is so important, It's expensive to make purines, especially purines, from de novo. It's too much ATP. So the process occurs in the cell in one big complex. So it doesn't go far, but it requires a lot of energy. All right, so we have IMP, we have inosine monophosphate. What do we do with it? Well, we could do one of two things with it. We could take that inosine monophosphate, so that's the IMP, and we could turn it into AMP, or we could turn it into GMP. So when we look at this, what we want to realize is that we are going to want to regulate this fork in the pathway, right? Because we have IMP and it can either go to AMP or it can go to GMP. So the products of each of the forks in the road are going to be regulators. So this is going to be a negative regulator for that pathway, right? So it's immediate feedback inhibition of the division in the pathway. And so that actually kind of forces not only you to stop one side of the pathway, but also to promote the other. Okay, so if you have inosine monophosphate, let's say you want to go and make AMP. To go and make AMP, you're going to have to add another methyl group. So where are you going to get that methyl group? You're going to get that methyl group from aspartate, and you need some energy to do it. But this is kind of strange, right? This is not ATP. We're using GTP, so you use GTP to make ATP. And then if I kind of look on the other side and just make a guess about what's going to happen here, right? I can see that I'm going to use ATP to make GTP. And so this is another level of regulation. We don't want to use ATP to make AMP. Like, that just doesn't make sense. So it's a substrate specificity so that we have that specificity, and we can require different energy sources in order to see that branching. So it helps us to balance our AMP and GMP levels. But okay, so we've got, we made AMP. So now we have to talk about making GMP. So this one, again, requires our energy, and we need to add a nitrogen group. In this case, we're adding our nitrogen group in a different part of the ring, but we're going to get that from glutamine and our input of energy, and we make our two monophosphates. Okay, so that's great, but in order for AMP and GMP to be useful in the cell, first we have to convert them to a nucleoside triphosphate. So we got to go through two phosphorylation events. So the first phosphorylation event occurs pretty much the same for AMP and GMP. So when that happens, we go from having one phosphate to having two, having one phosphate to having two. So it's either adenylate kinase or guanylate kinase. And for this, they both use ATP. So anytime you're just phosphorylating something, typically we use just ATP. So now we have the diphosphate versions of each of the nucleotides. And now we have to make a decision. The cell has to know, am I going to go and use this nucleotide to make RNA, or am I going to go and make it, use it to make DNA? If I want to make RNA, all I have to do is add one more phosphate group. So I go through another kinase. This is a nucleoside diphosphate kinase. Add one more phosphate group on it. I got ATP, and I am ready for RNA synthesis or whatever else I need to do. On the other hand, maybe I need some DNA. So I go through this reaction. Now, this is an extremely important reaction. I'm going to talk about it again shortly. But this reaction happens where we convert ADP to deoxy-ADP, where we remove the hydroxyl and we just replace it with a hydrogen. Through ribonucleotide reductase. This is going to be an extremely important enzyme, and we'll talk about it over and over again. So once we convert that to a deoxynucleotide. Now we do the next phosphorylation step, and this one happens with basically any phosphate, any trinucleotide that's got an extra phosphate. So then we have the D, ATP, and we can make DNA. Similar sort of thing happens for GDP. Either I go ahead and make RNA just by adding another phosphate, I got GTP ready to go for RNA, or I have to go and make DNA. So I go through ribonucleotide reductase, get rid of my hydroxyl, and then phosphorylate it for the second time, right? So you just have to remember this enzyme we're going to keep coming back to, that is so important when we convert this nucleotide from being used for RNA for being DNA. Okay, so we know feedback inhibition, right, is going to be really important. We've seen feedback inhibition for amino acids to make sure that we have the right proportions of each amino acid. And it's also important for nucleic acid biosynthesis, right? You need equivalent amounts of purines and pyrimidines. So remember, the way that your nucleotides pair, A always pairs with T. So A is a purine, T is a pyrimidine. And the way you remember this is there are apples in the tree. So A and T are always together. G and C are always together. That's cars in the garage. But no matter which base pair you're talking about, you're always going to have one purine, and then you're going to have one pyrimidine. So you're going to have equal or equivalent amounts of purines and pyrimidines. So if we kind of look at how this gets regulated, we have a couple of steps where this happens. So the very first point of feedback inhibition in purine biosynthesis is the PRPP synthetase. So PRPP synthetase is inhibited by both ADP and GDP. Right? It prevents that expenditure of the 2-ATP for me to convert my ribose 5-phosphate to PRPP. Because if I can recycle the PRPP that I already have in the cell, great. I don't want to waste that 2-ATP. The second level where we regulate purine biosynthesis is with our glutamine PRPP aminotransferase. Now, this is inhibited by lots of different things. Every different version of the adenosine and the guanidine bases, whether that's mono, di, or triphosphate, all of them. So you get independent inhibition because, like, any product of adenosine will give you inhibition, or any product of guanine. So you don't have to have both, but if you do have both, you actually get synergistic inhibition. So you have both independent and synergistic, which is really nice. It's very effective. And then the very last step where we have a good amount of regulation is where we have our fork in the pathway. This is another fork in the pathway, right? Where we're looking at IMP going to either ADP or, well, AMP or GMP. And so our AMP and our GMP molecules will come up and they will inhibit that conversion of the IMP to whichever nucleotide we're talking about. And so you have direct inhibition of the different forks of the pathway. So if you shut down one side of the pathway, you're going to increase flux through the alternative branch. So that's what helps us to balance our AMP and GMP levels. So that's really important. Okay, so we've made purines. We know how to do them. Now, what happens if we have to degrade them? Well, if we degrade them, we turn it into uric acid. And when you look at kind of how this happens, you're going to start with either AMP, IMP, or GMP. And so what we want to do is we want to get rid of the nitrogens, right? So the first thing that can happen is you can take AMP and you can deaminate it and you can turn it into IMP. So that's one way that you can get rid of just one of those nitrogens. But you can take any of these three nucleotides and you can dephosphorylate them. So when we're starting out, we have the sugar with the phosphate and we have the base. And so if we go through a dephosphorylation where we lose the phosphate group, what we're going to do is basically kick the base off of the sugar. And so I'm going to be left with just the base, whether that's adenosine, inosine, or guanosine. That's all we're going to be left with. Okay. So, and at that point, adenosine can be deaminated to inosine as well. All right. So now we're left with basically inosine or guanosine. So now we're going to release that sugar from the base, right? And we're going to be left with just the base. So these two are our bases. We have hypoxanthine and we have guanine. And so now we want to get rid of the nitrogen containing components, right? And so we start to do that and we get xanthine, right? So we go through one, this is a deamination here. We end up with xanthine, whichever way we go, and then we're going to convert our xanthine into uric acid. And uric acid it's a good waste product because it's insoluble in water. Like urea requires a huge amount of water in order to be soluble. But it can be used by birds and reptiles and insects and all that kind of stuff. So if you look, animals make uric acid, but we usually degrade it into other things, but different animals will degrade it to different points. And you don't really have to memorize that. I just want you to kind of see that each different animal can degrade that uric acid into different substrates to be secreted out in different ways and that kind of thing. So you don't have to spend a lot of time on that. So let's talk about what happens when something goes wrong, right? That's the fun stuff. What are they going to say about me? Anyway, so gout is an example of a metabolic disease of purine metabolism. And gout's really, really common. It's actually really common down here. And it's a problem in degradation of our purines. And so we know that we're going to degrade our purines into uric acid. So if we have too much uric acid, those uric acid crystals are going to build up, and they build up in primarily synovial fluid in our joints and in the kidneys. And so if you've ever seen uric acid crystals, here are uric acid crystals. They look like syringe needles, like they're extremely sharp and extremely painful to have. And so what you see is people who have gout, if they stub their toe or something, I mean, they are just about on the ground in pain because those kinds of blunt injuries release the uric acid crystals and give them inflammation. And I mean, it's just a pretty painful disorder. So how can you get gout? I don't like the way the book kind of arranged it, so we're going to arrange it a little differently. So gout is linked to one of three different defects in the degradation pathway. The most common is an increase in PRPP synthetase activity. So PRPP synthetase is the first step to go and make inosine monophosphate, IMP. So you end up making way more IMP than you need. So that's a way that you get it. You can get it if you have a defect in the feedback inhibition of the glutamine PRPP aminotransferase, ASE. Oh, look, ASE. And we're going to go and look at these on the pathway in just a second. And the last way is to have a defect in the salvage pathway. And so let's go and look at that, right? So I have those same three here. So the very first one, if we have an increased enzyme level of PRPP synthetase, Then I'm going to make more PRPP, which is going to make more of every single thing that's below it in the pathway, which ends up making uric acid. So then uric acid builds up and we get gout. So that's one way that it can happen. The second way is if your glutamine PRPP aminotransferase, which is supposed to be feedback inhibited by its products, if it's insensitive to that feedback, then it's not going to stop when it should. And again, your pathway is going to keep going. You're going to build up uric acid. That's going to lead to gout. And then the last way is if you have low levels of this enzyme HGPRT. HGPRT takes hypoxanthine and recycles it back to IMP. And so if you don't do any recycling, then hypoxanthine is going to build up, which means I'm going to make more uric acid, which means I'm going to have gout, right? So it happens in the past, in history, it's been called the disease of kings because it's associated with a diet that's really, really high in alcohol and meat. And kings were the only ones who could afford enough alcohol and meat to get gout. So that's why it's called the disease of kings, because the alcohol interferes with uric acid excretion. So if you can't get rid of it, you're going to have problems. And then the more meat you eat, the higher your levels of nucleic acids are because you're getting it from your diet as well. So then you're just increasing uric acid in multiple ways, right? So that's what leads to gout. So how do you treat somebody who's got gout? Well, you restrict their diet and you tell them you cannot eat purine-rich food. Purine-rich food is typically organ meat or glandular meat, bacon, turkey, I know, right? Anchovies, one of my favorites, sardines, right? You could give them all that. You could restrict all that. Or really it's probably you give them a drug called allopurinol. So right here, this one. So the allopurinol prevents that conversion of the hypoxanthine to the uric acid. So it's a competitive inhibitor of the xanthine oxidase. And so it prevents that last stage conversion of hypoxanthine to uric acid. But these are not, this gout is not the only disorder that you can have if you have a defect in this degradation pathway. There are much more dangerous disorders that you can have. They're less common, but they are way, way, way more dangerous. So if you don't have any ability to recycle your hypoxanthine into IMP, so your HGPRT just doesn't work, then you have Lesch-Nyland syndrome. So this one is extremely rare. And so you can't do that recycling thing. And so you have, the book doesn't say it, but you have gout symptoms. And then you have some weird stuff. You have these really weird neurological symptoms, severe anxiety and self-mutilation. It's a really strange combination. And we're really not sure why. It's under investigation currently. They think that it's possible that because your nucleotide levels are so low, because you're not doing the salvage pathway at all, you are depending on de novo synthesis completely, and it is so expensive for the cell that you have a really low pool of nucleotides. And so the brain doesn't develop the way that it's supposed to because it's a high energy using organ. And when it's growing, it's even more energy consuming, and therefore the function's compromised. But they're just not sure why. It's like anxiety and self-mutilation. I don't know. It's a kind of interesting, like, why. Okay, and then the last one, let me come back up real quick, is if you cannot take adenosine and break that adenosine down into inosine. So if you have a problem doing that conversion, it's right here, this conversion right here. So that means that you're missing the adenosine deaminase gene. This is really important. You can have what's called adenosine deaminase deficiency. It's called ADA and ADA leads to SCID. So what is SCID? SCID is severe combined immunodeficiency disease. So basically you have an immune system that doesn't work. But why? So if you have a defect in adenosine deaminase, you keep these really high levels of adenosine, and that's the sugar in the base. And so when that starts piling up in the cell's like, I got to do something with this. And so it ends up turning it into the deoxynucleotide, so the DATP. Well, DATP is an inhibitor of the ribonucleotide reductase. Pause, go up, and go find where that enzyme was. Right here, ribonucleotide reductase. This is the enzyme that allows us to make nucleotides that are required for DNA. So come back. Sorry about all the scrolling. So if we have a defect in the adenosine deaminase, right, and I make a ton of DATP, then I inhibit the ribonucleotide reductase. No other DNTPs are made. I can't make anything else for DNA. And so the DNA can't replicate and the cells can't replicate. And so the cells that are highly turning over very quickly, rapidly dividing, those are the most affected, which are the immune cells, specifically B and T cells. And so if you don't have B and T cells, your immune system is shot. I mean, it's bad. And so you see people who have this disorder, these they made a bad video of it, but like Bubble Boy, right, where you have to stay protected away from everyone else, you're in isolation, and it's to prevent you from having life-threatening infections. You can give the patient IV antibiotics, immunoglobulins, right, to help them treat, but it doesn't really do much. So the only real treatment is currently bone marrow transplants, and they typically like to do it from siblings to avoid rejection because they're genetically so similar. But there is an experimental treatment, and it's a gene therapy treatment. And so this is kind of a good place to explain how gene therapy works. So what they do is they take out a patient's hematopoietic stem cells, and these live normally in the bone marrow, and they give someone a drug, and that drug tells those hematopoietic stem cells to leave the bone marrow and go into the bloodstream. And so then they can just harvest it from the bloodstream. And they culture them, right? They keep them in a petri dish. And at the same time, they're also creating recombinant viruses. And so it's a virus that normally would infect human cells, but can't. It can't infect, it just can't replicate. And it doesn't have all of its normal genetic information. It has in its place the adenosine deaminase gene. And so it's a retrovirus. And so what happens is when the virus infects the cells, it actually inserts part of normally its chromosome into the human chromosome. But instead of inserting viral DNA, the researchers program it to insert the adenosine, the correct copy of the adenosine deaminase gene. And so you have these cells that get infected, right? And some of them become infected and some of them don't. So you can see that like this one and this one, both of these got the gene for the good adenosine deaminase, and the other two didn't. And so they select out those that have picked up the correct gene, and they put those back into the patient. And hopefully those still hematopoietic stem cells that now have a good copy will go ahead and recolonize the patient's bone marrow and have functional adenosine deaminase and be okay. It's really in the preliminary stages, but and you have to be really, really careful that you don't induce uh, cancer, because sometimes if you insert the gene in the wrong place in the chromosome, it'll either activate oncogenic genes or deactivate tumor suppressor genes. And so it can be really dangerous, and you don't want to do more harm than good to the patient, you know? Okay, so, and we're going to end there. All right.